So we're talking about this DNA sequence here. A point mutation might change this nucleotide here. Okay? That could have the effect of changing this entire protein, or not changing it at all. Okay? If it doesn't change it at all, it's a silent mutation. If it changes it slightly, or we get a missense mutation, what you can have happen there is that the incorrect amino acid, in this case, this one here, gets put in place. Okay? The protein might still be able to function, but not as well. We might call it a leaky mutation. In this case, the microbe maybe grows slowly. You could have a nonsense mutation. In that case, when you go back to our sequence here, a nonsense mutation might happen here, or here, excuse me, such that it changes this to a stop codon, and so the protein doesn't get coded at all. It just stops the transcribing of that particular gene. In that case, we'd call it a nonsense mutation because the protein doesn't get produced. You might also refer to it as a null or knockout mutation. Any questions?
Can you go over the base substitution again?
Okay, so base substitution is just, replacing one of the nucleotides in your sequence with a different nucleotide. So in this case here, we have an A here, but if I, in copying this nucleotide, accidentally put a T there, that is a base substitution. Okay? And it only happens at one point, so we call it a point mutation. And depending on where it happens, it could change the amino acid so that the protein still functions, but not as well as it did. So we get some slow growth, we call that a leaky mutation. It could change the sequence such that it puts in a stop codon, and so the protein doesn't even get produced. And we call that a nonsense mutation. So both a missense and a nonsense mutation are point mutations. They're base substitution mutations. Okay, but there's another type of base substitution mutation, and that is, well, not really a base substitution. It's an addition or removal of the nucleotide. So if you addition or removal of base, if you remove one of the bases when you're copying the DNA sequence here, and I'm going to go ahead and redraw this. Okay, so now I've got 3 nucleotides here. If a addition occurred right here, we put a G right in here, look what that does to our sequence now. We still have this amino acid, this triplet code, but now we have a different triplet code here and a different triplet code here. So it shifts the sequence. Same thing happens if instead of an addition, we have a deletion or removal. Now, you've still got this code, but now you have a different triplet code here and a different triplet code here. An addition of a nucleotide to the sequence or the removal of a nucleotide to a sequence causes a frame shift mutation. the whole frame, the whole sequence shifts when you add or remove a nucleotide. In most cases, a frame shift mutation is going to lead to a knockout condition. The protein is probably not going to function or not going to be produced. Every once in a while, though, it produces a novel protein, one that will fold up on itself and create or allow for a completely different reaction that the cell couldn't produce before. One more type of mutation that we want to mention here involves transposable elements. Transposable elements, segments of DNA that jump spontaneously. Okay, so again, here's our circular DNA for our microbe. A transposable element might be this sequence of DNA right here. And what happens is it excises itself out of this spot and jumps to a different spot. What this can do then, if you had a gene that was running, say you have a gene that runs from here to here, and this transposable element jumps and inserts itself right here, you can disrupt that gene. I know it sounds dirty, but it's not. Insertional inactivation is what's going on here. The transposable element is inserting itself and inactivating the gene product. Any questions?
So transposable element is just the gene that's jumping to this?
It's not even a gene necessarily, it's a segment of DNA, a region of DNA that jumps. And it doesn't necessarily have to even code for a protein.
So what do you mean by disrupting the gene? So it stops the production of amino?
It stops the production of the protein. So messenger RNA is not going to be transcribed, and the protein is not going to be translated.
Is that a totally random event, or is
Yeah, it seems to be. Yeah, as best we can tell. So how can we produce mutations? Chemicals are effective in inducing mutations. We call them chemical mutagens. So a lot of times they can alter the binding between the hydrogens of the bases, interfere with that. They can modify the purines and pyrimidines, those are the bases as well. We can use this notion of the transposable elements to induce mutation, inserting transposons that then will jump to cause the mutations. And as I said, usually it results in a knockout. So if you're trying to shut down a particular gene, if you can get the transposon to jump into that gene, you can shut down that gene. Radiation. Another way of inducing mutation UV radiation causes mistakes, causes basically dimers to form between the thymine molecules, and then these have to be clipped out and repaired, and in the repair process, mistakes are made. So radiation, UV radiation, can cause mutations to occur. So what are the consequences of a mutation? If you think about this, then that means that not all daughter cells are going to be genetically identical. And I keep kind of saying this when we talk about a colony. You know, here's our petri dish, and we've got a colony on there, isolated. And I'm telling you right now that we can establish a pure culture from this colony because all the cells are genetically identical. But they're not, are they? If you consider a mutation, and at the rate that mutations take place, we can be safe in assuming that some of these probably have undergone some mutation. So if we could do a genetic sequence of every cell in this colony, we would see that there are some differences in those nucleotide sequences. They're not really all genetically identical. But we make that assumption, otherwise we could never establish a pure culture. And for the most part, they probably are. Because like I said, many mutations will lead to nothing. They'll either be silent or they'll be knocked out. And so they don't even get passed on to the next generation. It is rare that a mutation provides some kind of advantage that does get passed on to the next generation. But once a mutation takes place, now natural selection can serve as a force on that mutation. The environment will influence whether or not that mutation is going to survive. So does that mutation provide an advantage to the cell? And if it does, then the mutation is going to survive and be passed on to the next generation. The advantage will allow that microbe probably to dominate others. Resistance to drugs is one of the ways that mutations help microbes take advantage of their situation. So let's move on to gene transfer now. So all this talk about mutations happening in the DNA sequence and then gets passed on to the daughter cells. And that being a vertical transfer. Now we're going to look at a horizontal transfer. Gene transfer, and there are really three types of gene transfer. And we're talking about now the transfer of genes from one bacterium to another bacterium. DNA-mediated transformation or transfer is what happens when a cell picks up DNA that is freely floating in the environment. Picking up naked DNA, if you will. Some cells have a natural competence for this. When a cell dies, or when a cell lyses, the stuff that's in that cell gets released into the environment, including the genes of that cell. Some cells have the ability, through pores within the membrane, to bring that genetic material into the cell. Sometimes in doing so, they gain gene characteristics that they didn't have before. We can induce this as well. Take some cells, put them in an environment where there's a bunch of naked DNA, and then shock them. With the electroporation, they can punch holes into them. And so now you've got holes in the cell membrane, and that allows the DNA to move into the cell. So if we wanted to introduce a particular gene to a cell, we could use this method of artificial competence. Punch holes in the cell membrane, and let the DNA move into the cell. Now, of course, not all cells are going to pick it up. And maybe they don't all pick up the right gene or the right sequence. Transduction. We talked about transduction briefly when we were talking about viruses. Viruses carry out transduction. Phage have the ability to excise themselves out of a DNA sequence and take some of the host's DNA with it. Let's go back over here to so again, here's our bacterial sequence, and a virus infects the cell, a phage infects the cell. I need more color chalk, I'm sorry. And it inserts itself right here, okay? So this phage inserts itself in this DNA sequence. Now, when it gets ready to leave, excise itself out, it does so, but maybe incorrectly. And it takes some of the host DNA with it, leaves some of its DNA behind. In doing so, when it goes to infect the next bacterial cell that it infects, it's going to bring some of this host DNA with it. Two types of transduction.